Welcome you all to the more or less the last class in this particular course, but I will continue with the tutorial aspects that we have been discussing in the past 3 classes or so, and then I will sum up the whole thing. So, let us look at the queries that are shown over there with under the zinc in biological system. I have completed up to the copper in the previous class. Okay, in spite of the fact that the zinc does not undergo redox, that means zinc 2 plus does not go to 3 plus, does not go to 1 plus. Okay. It is involved in a variety of enzymes including oxidoreductase. So, there is a lot of enzymes are known, we know that, hydrolytic type, redox type. It does not undergo redox, but the reaction product has oxidation reduction. How is that possible? So, how is that possible? Because it does not undergo redox, but it will utilize the help of another cofactor, for example, cofactor of NADH. So, NADPH, which can act as a reducing agent, and that will help the zinc. So, that means the zinc active center and then NADPH center should be close in proximity and should get triggered together. All that requires that one. For that you have enzyme has a mechanism. Therefore, the zinc is involved even in oxidoreductases in spite of the fact zinc itself does not undergo redox. Let us look at the next one. In most of the zinc enzymes, there are one or more water molecules are always bound to the metal center. This is true. You look at any zinc enzyme, you always find either one or more, and there are some more waters close by also other than the coordination, but we are talking about the ones which are at the coordination. So, what do you think the role of such a water molecules? So, why these waters? Generally speaking, when you have a metal ion and having a water molecule on that, if a substrate comes in straight away, the substrate would more or less displace this water molecule. So, that means in the resting enzyme, in most of the cases the water is present, and when the substrate comes, the water will go away. Or in some cases this water gets activated, for example zinc, and that activation is supported by a general base like carboxylic group which is sitting close by, which will abstract a proton from your OH2 which is bound to zinc. Therefore, you will form zinc hydroxy. So, the most common things are either to replace the coordination by an incoming molecule or to convert the water into hydroxide which is a nucleophile. Now, this is most, in fact that is true in many other cases as well. In many enzymes the metal center may not activate the water to OH, but they use it as the position to be filled in for the place of the substrate. So, when the substrate comes, that will vacate. So, in other words, the substrate displaces this particular water molecule, and this is what happens even in zinc enzymes too. Let us get to the next question. How does the enzyme carbonic anhydrase converts the CO2 into bicarbonate? So, obviously, to convert CO2 into bicarbonate, you have to add one more OH minus. So, this is clue has come already. So, what is the mode by which it binds to the zinc center, and explain how one establishes this binding mode unambiguously. Say for example, let us say the carbonic anhydrase, now the CO2 comes. Either CO2 has to bind to the zinc center, or it has to be in the close proximity so that the zinc center can do some kind of a reaction. In fact, in case of the carbonic anhydrase, what happens is the CO2 does not directly bind to the zinc center initially. The CO2 as such is recognized by the protein, and it holds it is held by certain hydrogen bond interactions in such a way that the zinc or hydroxy is in close proximity to the carbon center of the CO2. Therefore, the carbon center of the CO2 gets attacked by the OH. So, that is electrophilic center is the carbon, the nucleophile is OH. So, zinc OH attacks at this, and as a result of that it will bind zinc O, C, O, that O also makes with the zinc another transition state. So, you have a transition state is being formed, and this breaks down in presence of the water and gives the carbonate ion. So, what is required? Carbon dioxide should be recognized, carbon dioxide should be held in the close proximity to zinc and in the proximity of the zinc hydroxyl species. So, that is how it happens. Okay, let us look at another question. How does alcohol is converted to aldehyde product by the enzyme alcohol dehydrogenase? So, this is the again dehydrogenase, alcohol dehydrogenase. This is what we were referring to, oxidoreductase. So, obviously directly the reactivity is not happening as a zinc redox, but zinc helps in one binding alcohol, also activates in the neighbor residue the NADH. Therefore, the reaction occurs and oxidation takes place. So, what happens to human if this liver was not, if this enzyme was not present in their liver? Obviously, we know that when say somebody takes alcohol, we say, oh, your liver gets affected. That means we know that it goes into the liver, and the liver where the reaction occurs. So, if this enzyme is not there, the alcohol will not be converted to the corresponding aldehyde. So, obviously, alcohol remains as alcohol, which is dangerous obviously. So, that means one cannot consume alcohol if this enzyme is not present in their body, in their liver. So, obviously, it is required to have the alcohol to be utilized. Let us look at one more query in this context of zinc in biology. In the family of carboxypeptidase enzymes, what is the term carboxy refer to? So, the carboxy here refers to the carbonyl part or the carbonyl end or the C terminal part of this. In this family, there are several enzymes such as carboxypeptidase A, B, and glutamate carboxypeptidase, so many things are there. So, why so many different enzymes are required? Why all of these are? Yes, no doubt, all of these are peptidases, but the enzyme has to recognize that particular peptide bond. So, different kinds of the peptidases will recognize different types of the peptide bonds. What do you mean with type of peptide bond? All peptide bonds are same, but the one carb C alpha on the left side, one C alpha on the right side have got different R groups or same R group. So, therefore, that R group is the one which is differentiates. Suppose you have a aromatic moiety on this one, then a carboxypeptidase A will go. Okay. So, like that you have a negatively charged, you have a positively charged, you have a hydrophobic kind of thing. So, based on that different kinds of peptidases are required. So, therefore, the nature has developed variety of family of carboxypeptidases, and these are all the ones which work based on the zinc. Okay. So, okay, let us move on to the topic on the molybdenum. Okay. So, the molybdenum, okay, let us see some queries on that. Justify the dominant bioinorganic role of molybdenum. Okay. Justify the dominant bioinorganic role of molybdenum that is played as compared to the chromium, okay, where chromium does not involve in any of an enzyme. Why the question arises? Is the chromium, molybdenum, tungsten, they are in the same family. So, if they are in the same family, why cannot one think that the whatever the molybdenum is doing, why chromium has not done? It is a very interesting kind of thing. So, what do we need to think of? We need to think of, because all these reactions in the molybdenum are happening by their redox chemistry, by their oxophilic chemistry, by their oxotransfer chemistry. Now, if you use all these 3 points for molybdenum, all these 3 points for chromium, you will see the difference that the chromium does not have a suitable redox potentials as that of molybdenum, because in the enzymes, as you will see later on, the enzyme will go from molybdenum 6 to molybdenum 5 to molybdenum 4. Similarly, chromium also has to go to chromium 6 to chromium 5 to chromium 4 and it has to be stabilized, and the redox potentials have to favor. Also in the process, oxygen should be added to the substrate, oxygen should be taken away from the substrate, which cannot happen by the chromium, is not so oxophilic as that of the molybdenum. So, because of all these reasons, the bioinorganic chemistry of molybdenum is more in the nature than that of the chromium, and in fact no chromium is found still in any enzyme at all, because very strong reducing, very strong oxidizing these characters of this chromium would be a disfavorable factor for itself not to be in the enzyme. Let us look at the next question. Write the totally balanced reaction for the conversion of N2 by the enzyme nitrogenase. Of course, here you have to take not a nitrogen reaction in a test tube, but the nitrogen reaction by the enzyme. Though the nitrogen requires 6 electrons 6 proton, the enzyme requires 8 electron 8 protons, and it gives not only 2 NH3, but also gives H2, and this hydrogen comes out first and the enzyme is activated, and then only N2 can get activated, otherwise it will not get activated. So, therefore, total reaction by the enzyme must have both 8 electrons and 8 protons. So, you can refer to my slides and the lecture that I have given you earlier. Let us look at the next one. Draw and depict the highlights of the species of conversion and N2 by just focusing at what has been known to happen at the iron and molybdenum centers of the iron molybdenum cofactor in nitrogenase enzyme. The nitrogen requires totally, this enzyme requires 8 electrons 8 protons. So, therefore, you have each step, and first 2 electron 2 protons will go as H2, then the enzyme is ready to accept the nitrogen, and the N2 will be reduced by 1 electron, 2 electron, 3 electron, 4 electron, 5 electron, 6 electron, and correspondingly goes through all these intermediate steps, and then you have the final ammonia. And there are different kinds of mechanisms which I have given in the class, you can please look at that. At what stage does MO nitrogenase take its active form? I just now explained, after the H2 is evolved. What is the balance reaction for the nitrate reductase? Nitrate to nitrite, nitrate to nitrite kind of thing. So, it is basically a reduction. So, correspondingly the electrons are taken in and one of the O goes as water. How does this happen at the enzyme active site? Catalytically, because initially the nitrate can bind to the molybdenum center, and then the molybdenum center undergoes redox. How many redox electrons are required here? 2 electrons. So, molybdenum 6 will go to molybdenum, molybdenum 4 will go to molybdenum 6, and the nitrate goes to nitrite. And then the enzyme which is gone to 6 has to be brought back to 4, and that is done by another system of enzymes like cytochromes. Let us not worry about that at this stage for this question. Okay, in the enzyme xanthine oxidase, secondary coordinations interactions from the protein are extremely important. Why? Yeah, they are extremely important because they have to recognize the substrate. When they recognize the substrate, they will also favor its orientation, and if they are favored in orientation, the reaction can take place properly from the molybdenum center to that of the xanthine part of it. Let us look at some other also on the molybdenum. Simple dioxo molybdenum 6 complexes goes into a thermodynamically stable species and hence get prevented from completing the catalytic cycle. What are such species, and comment on how such species prevents the catalytic process. What happens if you take a simple dioxomolybdenum and add some reducing agent with 1 electron, it will go to molybdenum 5, but such species, 2 of such molybdenum 5s can join together to a oxomolybdenum. Just like I talked to you when in case of the iron oxygen bound species when reduced can get the iron oxo species. Similarly, molybdenum oxo molybdenum with molybdenum 5. Once that is formed, it is irreversible. Therefore, it prevents the catalytic cycle. So, what needs to be done? The enzyme must be smart enough that when it comes to the molybdenum 5 form, no two molybdenum 5 units should come closer. That means protein should cover it up and they will not form any kind of a dimer or mu oxodimer. Okay. So, the species is the mu oxodimer molybdenum 5, and you can easily find out this by the EPR spectroscopy, etc. How much such species prevent the. Yes, once they are formed MOO, which is molybdenum, oxomolybdenum with a bridged oxygen oxomolybdenum, and this is the molybdenum 5 for each. So, this molybdenum 5 is an irreversible species, therefore, catalysis is basically stopped. Okay, let us look at some queries in the last part of the enzyme that I talked to you on the mercury and talked to you on the selenium, etc. Okay, what is the mercury reductase? So, mercury reductase reducing the mercury, reducing mercury from mercury 2 plus to mercury 0. What is the impact on the environment? The mercury 2 plus in fact is a more dangerous species for the biological system. Mercury 0 is somewhat much less danger to the system. That is why the mercury 2 plus toxicity is being reduced to mercury 0. In fact, this happens by enzyme called mercury reductase. This is present in a large number of microorganisms. The microorganisms obviously are developed even in the environment. So, in the environment, if you can create certain kind of a stimulation or the nutrients, these microbioles can grow, and these microbioles will generate the enzyme called the mercury reductase, and that reductase will reduce mercury 2 plus to 0. How does this function? This enzyme is, of course, is a complicated enzyme, but this enzyme works mostly on thiol functions. Reduction is called cysteine, oxidized cysteine is called cysteine or disulfide. So, there is a chemistry play between the cysteine and disulfide and cysteine. Cysteine is a reduced form, disulfide is oxidized form. So, between these two, and that is how it happens. Okay. So, therefore, it is mainly play. So, initially when mercury comes, there are some cysteines which will take up this mercury, but at that stage it will not happen. That will transfer to another site in the enzyme, as I explained in the class. You can see the slide for that structures, and from there where the FAD and NADH units are close by, which will reduce the mercury 2 plus to 0 and oxidize the thiol functions into disulfide. And then that disulfide is again broken back into the thiol, so that your cycle is starting. So, this is how it functions in the biological systems. Okay, what is the form in which selenium is present in the body? The selenium is not present as a selenium 0, is present as a organoselenium, which is called selenocysteine. So, instead of sulfur, assume Se, everything else is same, and that kind of things. You know, SS can bond to disulfide. Selenium similarly, selenium can also bond with selenium with sulfur, selenium sulfur disulfide, selenosulfide kind of thing. So, it can undergo whatever is undergone by the sulfide. So, therefore, this kind of reactions occur in the selenium enzymes where redox can occur as a selenium, and selenium with hydrogen is called selenol, just like thiol. When it has a hydroxy group SCOH, it is like equivalent to carboxylic kind of thing. So, it can undergo all of these, and as a result of this enzymes called glutathione peroxidase and thioredoxin reductase, and these all where the selenium part of the enzyme is involved. See, I have gone through a large number of cases and it is idea of this tutorial is not to just give you the answers, but give you the approach along with the answer, or approach and ask you to look at the slides, and this is what I have done. So, do not think that for every case I give just the answer. No, that is no use, because then that means you have to mug. You should not mug, you should try to make yourself understand the subject. So, in effect what I have done? Was, in the entire course I have done initially the teaching. The next part of that, in the teaching, at every stage, every topic I have made a conclusion. For every super topic also I have made a conclusion kind of thing, and try to compare at each stage. It is a comparative way I have always been teaching. Okay? So, therefore, that is where I think it should be very stimulating things. Subtopics I have concluded, super topics I have concluded and correlated also. Then at the end I have given something for 3-4 hours, I have given the highlights, which is again concluding every chapter that you studied earlier. Followed by that I have given last 3-4 hours the tutorials as how to address the question. I have told how to answer the questions by using the question itself, by using our understanding of the thing itself. First we should coolly read the question, try to understand, try to see what with respect to the enzyme, what with respect to the chemistry point of view. If you club all of these, it will become the biological inorganic chemistry or inorganic chemistry for life. Thus, I have done the initial teaching part, teaching with the comparison to correlations and conclusions at every stage, super stage at the end of the thing. Then towards the end I have also covered a large number of the tutorial problems, given the approaches how to look at this problem, how to make. Of course, I have given the answers also in more or less almost every case except few where you need to look at the exact slide for you to understand, etc. So, in the process I have done a lot of introductory for 6-7 hours or so, which covers the general rules of elements in biology, then some coordination chemistry features, a lot of biomolecules, protein synthesis kind of things, variety of techniques, all of these I have given. Then I have taken aspects relevant to the alkali earth ions, biological perspectives, their biochemical aspects, their binding strengths, and their binding versus transport relations. They transport things in the ATPases, all those things have been. Case of the calcium, the trigger functions are also being. Then I took a host of enzymes, transition metal based, starting from titanium, because there is no enzyme, vanadium there are enzymes, chromium there are no enzymes. So, therefore, I have not taken manganese, there are a lot of enzymes, iron there are large number of enzymes, cobalt large number of only one enzyme, several enzymes, but one cofactor, that is vitamin B12 does a lot of reactions. Then nickel, lot of different enzymes, and copper, lot of different enzymes, zinc also lot of different enzymes. Then I have gone to the molybdenum, absolutely large number of reactions. Then I have talked to you mercury reductase, then I have talked to you selenium kind of thing too. So, overall the more or less that is required to this. Then I have talked to you environmental detoxification, that is mercury reductase. Then I have talked to you a non-metallic element like selenium present in the glutathione peroxidase, etc. Then I spent 3-4 hours or so on the inorganics in medicine, maybe even more, and their disease diagnosis and therapy aspects, and thoroughly compare their effective things, etc. So, overall I try to build this course to suit inorganic chemistry of life, not just the bioinorganic chemistry, not just the biological inorganic chemistry, but inorganic chemistry of life, but at the level of principles and perspectives, because this is mainly targeted for masters, but as I said, even the third year BSc students can be utilized, and a lot of PhD students it will be very useful, thought provoking, and they can try to work on their research problems much more focused way. Before I end the last lecture, let me express my acknowledgements. See, this whole task has been possible by the help of a large number of a large contingent of people. First and foremost is the NPTEL group, the NPTEL as a whole. I thank them, the office staff as well as the staff present at the studio who have made all these things easy possible in the time frame that we planned. And more than this, I also need to acknowledge my course associates. I had 2 course associates who also happen to be my own PhD students who are working in this similar area of research which concerned with this particular lecture series, and they are the Ashiv Narula and Sirlita Polopelli. I thank them very much for all the kind of support that they extended throughout this recording of all these lectures, making all the slides possible in a nicest form that it is. So therefore, with this I would like to thank all the NPTEL and my course associates, and more than that I thank you all for taking up this particular course on inorganic chemistry of life, principles and perspectives. Thank you.